Now we'll discuss the next SAQ, that is SAQ 17, which is write short notes on restriction enzymes. We have to discuss about restriction enzymes. These restriction enzymes belong to a group. They belong to a group called as nucleases. They belong to a group called as nucleases, wherein nucleases are of two types. They are exonucleases and endonucleases. Exonucleases and endonucleases. When we are discussing about these restriction enzymes belong to the large group, that is nucleases. Nucleases are further classified into two types. They are exonucleases and endonucleases. Coming to that of the first one that is exonucleases, and the second one that is endonuclease. What do they do? What is the function of these exonucleases as well as endonucleases? Coming to that of exonucleases. These exonucleases, they remove nucleotides. What is the function of exonucleases? They remove nucleotides from ends of the DNA molecule. Exonucleases are the enzymes which remove nucleotides from the ends of the DNA molecule. Then what are endonucleases? Endonucleases are also the enzymes, but they cut the DNA molecule into segments. They cut the DNA molecule into segments or parts. They cut the DNA molecule into segments. So that's what. So restriction enzymes belongs to a large group, that is nucleases. Nucleases are of two types, exonucleases and endonucleases. Exonuclease, that is exit, simply we can say. Exonuclease, they remove the nucleotides from the ends of the DNA, whereas endonucleases, they cut the DNA into segments. That's what. And first of all, I can say the first and usually the endonucleases which cut the DNA into segments belong to the category that is restriction enzymes, which belong to the category that is the restriction endonucleases enzymes. First restriction endonucleases. Okay, first restriction endonuclease or first restriction enzyme was discovered in, or it was first isolated. First restriction enzyme was isolated in or isolated from Haemophilus influenzae. Haemophilus influenzae. Okay, so as we are discussing that endonucleases are the enzymes which cut the DNA into segments. So before going to the first restriction endonuclease enzyme, must also know that actually first of all there are two enzymes which have been isolated from E. coli. There are two enzymes which have been isolated from E. coli. E. coli is the bacterium. It is Escherichia coli. These two enzymes have been isolated from E. coli, in which what have these two enzymes? What are the functions of these two enzymes? One of the enzyme has added methyl group. One of the enzyme has added methyl group to the DNA molecule. One of the enzyme has added methyl group to DNA molecule, whereas the other enzyme has cut the DNA at specific sequence, that is of 6 base pair sequence. Okay. So, 2 enzymes have been isolated from E. coli. These 2 enzymes which have been isolated from E. coli, they have found to be having inhibitory effect, or rather restricting effect on the growth of the bacteriophage. Okay, got it? So, the 2 enzymes which have been isolated from E. coli have been found to have inhibitory effect on, or have been found to restrict the growth of the bacteriophage. The 2 enzymes which have been isolated from E. coli are, one of the enzyme have been added methyl group to the DNA molecule, whereas the other enzyme have cut the DNA into different segments at a particular sequence. That particular sequence which consists of 6 base pairs. This sequence is called as, what is this particular sequence called as? This sequence is called as recognition sequence. This sequence is called as recognition sequence. Okay. So, the first restriction endonuclease enzyme which was discovered which has been first discovered, or rather it has been isolated. It has been isolated from the microbe that is Haemophilus influenzae, and this enzyme, restriction endonuclease enzyme, which have been isolated from Haemophilus influenzae, is named as HIN2. The word H refers to the Haemophilus influenzae. Okay, Haemophilus influenzae, HIN2. Okay, this HIN2 specially, or this restriction enzymes especially attaches to the recognition sequence and cuts the DNA into segments from that particular recognition sequence. That means here it is very much clear that restriction endonuclease enzyme it binds to the recognition sequence and cuts the DNA into bits or fragments or segments, whatever it is. Okay. So, apart from cutting the DNA into number of segments, this restriction endonuclease enzyme also, it first inspects, or rather it checks the length of the DNA molecule. So, we can say that restriction endonuclease enzyme, it plays a dual role or dual function. That one is that it first checks the length of the DNA molecule and then binds to the recognition sequence, wherein it cuts the DNA into fragments by binding to the recognition sequence consisting of 6 base pairs. So, that is what the nature or the dual nature or the work which is performed by restriction endonuclease enzyme. Okay. Here we are telling continuously, I am going on telling that recognition sequence which consists of 6 base pairs. What is this recognition sequence, or what is it? Okay. So, here if we go with, we must also be able to know about, we must be also aware of one word that is, or one term that is called as palindrome. The term called as palindrome. What is this palindrome sequence? Palindrome sequence is that which can be read similarly on both the strands in 5-3' direction. For example, we can go with that of, this is the 5' sequence which is having the sequence like this. And this is of GAATTC. Okay, so this is the palindrome sequence, we can say, palindrome. Palindrome sequence can be defined as the sequence which can be read similarly on both the strands in the opposite directions, that is especially in 5-3- direction. 5-3- direction. So that's what about the palindrome sequence, wherein here the restriction endonuclease enzyme, it checks the length of the DNA molecule and binds to that of the recognition sequence, which consists of the 6 base pairs, and then it cuts the DNA molecule into segments or into fragments. So where does it cut again? Here, this restriction endonuclease enzymes, it cuts the DNA, or as I have just now said, that restriction endonuclease enzyme checks the length of the DNA and binds to the recognition sequence, where it cuts the DNA at especially what is the backbone of the DNA, that is sugar phosphate backbone, where it cuts the DNA in the sugar phosphate backbone slightly away from that of the palindrome sequence. Slightly away from that of the palindrome sequence, but exactly between the two same bases on the present on the both the strands. For example, the restriction endonuclease enzyme, which is going to act or cut between these two nitrogenous bases, that is between the guanine and adenine in the lower strand, as well as the same between the two nitrogenous bases, that is the guanine and adenine in the upper strand, same in the same. Okay, so that's what. So here the restriction endonuclease enzyme, by checking the entire length of the DNA, getting attached or binding to that of the recognition sequence, it cuts at the region of the sugar phosphate backbone, cutting the region of the sugar phosphate backbone, wherein a little bit away from that of the palindrome sequence, but just between the two, say, nitrogenous bases, same nitrogenous bases which are present on both the strands, between the two nitrogenous bases which are present on both the strands. Here I have given out with the in between guanine and adenine. Here also on in between guanine and adenine. So the cutting takes place like that. This cutting, which has been performed by the restriction endonuclease enzyme, leads to the formation of cohesive or sticky ends. Leads to the formation of cohesive or sticky ends. The formation of cohesive or sticky ends, it activates or it helps or facilitates, rather I can say, okay. The cutting of the restriction endonuclease enzyme leads to the formation of the sticky or cohesive ends, which will facilitate the activity of the enzyme, that is DNA ligase. Which facilitates the activity of the enzyme, that is DNA ligase. These restriction endonuclease enzymes are popularly or well known or well used under recombinant DNA technology. It is of very great importance under recombinant DNA technology. So that's what we have discussed about the restriction enzymes, which play a very important role in checking out the length of the DNA, and combining or rather attaching to that of the recognition sequence, cutting the DNA in the sugar phosphate bone, just bit away from that of the palindromic sequence, but exactly between the two nitrogenous bases present on both the strands, finally leading to that of the formation of the sticky or cohesive ends, facilitating the activity of the enzyme DNA ligase. Ligase always ligates, ligation process. Ligation is nothing but joining process. So that's what we have discussed. We have finished discussing about the short answer, that is 17 short answer regarding the restriction. Now we will discuss the last SAQ, that is 18th SAQ. The question is, list out the beneficial aspects of transgenic plants. The beneficial aspects of transgenic plants. So what are, first of all, we must know what are transgenic plants. The word is giving out here, transgenic plants, wherein the genes have been transferred into. What for the genes have been transferred? So for example, we can say that if at all a gene which is responsible to carry out or to bring about the best results in the sense, usually in the crops what do we need? The best nutrition value or the best yield or the best insect resistant or the best pathogenic resistant. So if at all genes which are responsible for these best characters are transferred into a plant, that is, these plants which have been received the gene which is responsible for all the best characters are called as transgenic plants. These transgenic plants are going to have all the beneficial characters. So what are the beneficial aspects of these transgenic plants? The first point if we go with is that transgenic plants which are resistant against pathogens and pests. So transgenic plants which are resistant to or resistant against, also we can say, pathogens and pests, resistant to pathogens and pests. So for example, if we go with, there are number of examples we can go with. One is papaya. Okay. So transgenic plants which are having the beneficial aspects, that is, one of the beneficial aspect is that transgenic plants which are resistant against pathogens and pests. The first example is papaya. Transgenic papaya is resistant against ring spot virus. Transgenic papaya is resistant against ring spot virus. Then the next one if we go with, that is BT cotton. It is also a transgenic plant. It is resistant against insects. BT cotton is resistant against insects. And if we go with another example, that is transgenic potato. Transgenic potato. The word here I am putting in a short form, that is T. That word refers to the transgenic potato. Transgenic potato is resistant against the fungus. That is Phytophthora. Resistant against the fungus, that is Phytophthora. Then transgenic tomato. Transgenic tomato is resistant against the bacterium. The bacterium, that is Pseudomonas. Bacterium, that is Pseudomonas. Okay. So the transgenic plants which are resistant against different pathogens and pests, examples are papaya, which is resistant against the ring spot virus, and BT cotton, resistant against insects. Transgenic tomato, resistant against the bacterium, that is Pseudomonas. Then transgenic Potato, which is resistant again, is the fungus that is Phytophthora. So that's what. Now coming to that of the next one, that is the transgenic plants which are resistant or which are made helpful or rather useful for food processing technology, which are made useful for food processing technology, which are made useful for food processing technology. Okay, so transgenic plants which are made useful for the food processing technology are, one, we can go with that of the variety of tomato which is named as flavor saver, So the transgenic tomato which is made for helpful or rather useful for food processing technology is flavor saver variety. What is this importance of this flavor saver variety of tomato? Flavor saver we are telling. Okay, so what is this importance is that it is highly resistant during the transportation, okay, as well as in storage process. Okay, so it doesn't spoil, it doesn't get spoiled, or rather you can also say it is bruise resistant. What is meant by bruise? Bruise is nothing but getting wounded. Bruise is the wound, you can say. So the transgenic tomato which is helpful in the food processing technology is called as the flavor saver variety, wherein it is bruise resistant. Bruise resistant in the sense it is resistant or rather is very much helpful during it doesn't get damaged during the storage as well as transportation. Why? Because of the delay in ripening. Because of delay in ripening. So that's what. So flavor saver variety of tomato is bruise resistant because it doesn't get damaged during storage and transportation due to its delay in ripening. So that's what one of the point we can say under food processing technology. Now the transgenic plants which are useful in improving the nutritional quality, A variety of rice, you can say that is a Japan variety of rice which is called as Taipei variety. Taipei variety of rice. This Taipei variety of rice, in which actually we all know that rice is rich in carbohydrates as well as the husk or the outer layer of the rice is having rich in vitamin B. So this Taipei variety of rice which is also called as golden rice, is made, or rather this crop is enriched with vitamin A. It is enriched with vitamin A, and this is now called as Taipei variety or golden rice in order to prevent blindness. So that's what the importance of the transgenic plants in improving nutritional quality. For example, Taipei variety golden rice which prevents or which is enriched with vitamin A which helps in preventing blindness. Now coming to that of the next one, that is transgenic plants which are useful in the hybrid seed production. This is the another one, hybrid seed production. Here the transgenic plants, for example, you can go with one of the example over here. Transgenic plants like Brassica napus. Brassica napus. Here male sterile plants are produced through this transgenesis. Okay. So male sterile plants are produced in case of Brassica napus, wherein which prevents, or rather we can say which avoids the risk of manual emasculation, which avoids the risk of manual emasculation. So directly we can say that male sterile plants of Brassica napus are produced through this transfer of the genes, wherein which avoids the manual risk of emasculation. We all know what is emasculation. You all have studied in your junior inter, that is removal of anthers from the bisexual flowers without disturbing the rest of the other parts of the flower is called as emasculation. And which, so what is it useful for? What is it useful? If at all male sterile plants are produced by this technique in case of Brassica napus, which is useful in reducing the cost of production of the hybrid seeds, which reduces the cost of the hybrid seed production. So this is one more point we can go with, that is hybrid seed production. So as well as hybrid seed production, there are also some more beneficial aspects, not only the hybrid seed production. Okay, so but also some of the transgenic plants are developed in order to resist against themselves. That is fifth one we can go with over here, that is resistant against chemicals, cold, heat, drought, salt, etc. Simply we can say that these are almost all the abiotic factors which have influence upon the plants. Okay, so transgenic plants are produced which are resistant against the chemicals, cold, heat, drought, salt, etc. For this, under this example, we can go with that of two examples, that is Basmati variety of rice. Basmati variety of rice which is made resistant against both the biotic as well as abiotic factors. That is one example under this. And one more example we could go with is that Roundup Ready soybean variety. Roundup Ready soybean variety. So Roundup Ready soybean variety is an herbicide resistant plant. So that's what. So the last one is that here the transgenic plants which are made resistant against chemicals, cold, drought, salt and the heat conditions. Two examples we can undergo with this is that Basmati variety of rice which is made resistant against both the biotic as well as abiotic factors. Whereas there is one more variety of soybean which has been developed, that is transgenic variety of soybean, that is named as Roundup Ready soybean variety. What is it? Roundup Ready soybean variety of soybean which is made herbicide resistant. So these are the 5 most important beneficial aspects of transgenic plant. So we have come to the end of discussing the short answers, that is all the 18 questions we have finished.